right, our next speaker is Gloria Brunori. So she actually represents my lab at University of Florida, UF Scripps, and she's gonna tell us about modeling, something about modeling, mouse, GNAO1, G203R.
All right. Good afternoon, everyone. It's really my great honor to be here today and really witness in person so much progress and research and involvement in this GNAO1 community. Today I'm going to present my most recent progress on this project. And I'm trying to not to spend too much time on the introduction. We all know GNAO1 is the gene encoding for the G-alpha protein subunit, which among the 16 different alpha subunits expressed in mammals stands out as the GPCR signaling transducer with the highest expression in the brain. Many labs have performed G-protein coupling profile of GPCRs, and this is our most recent profile showing that of over 100 GPCRs analyzed, as many as 80% of them are able to couple to GO. So we are definitely dealing with a major transducer of brain signaling, and this was also emphasized and evidenced by earlier studies in mouse lacking the expression of the GNAO1 gene, which show very high mortality early on, and even the few mice surviving show high mortality rate in the very few first few weeks of age. The surviving pups also show severe body weight reduction and a series of neurological dysfunctions that manifest as you can see in the bottom panel as hyperkinetic behaviors and these sort of turning movements in the open field. You all know GNAO1 came into clinical attention in 2013 when multiple heterozygous de novo mutations were found to be causal to early infantile epileptic encephalopathy. And since then, many more mutations have been identified in patients with movement disorders, epilepsy, and neurodevelopmental delays. GNAO1 patients show different combinations of these symptoms and different degrees of severity ranging from mild to severe. And this is likely due to the fact that the 50+ pathogenic variants identified so far distribute all along the length of the G protein O subunit, and with a subset of these variants localized to highly conserved regions of the protein that are important for its function, such as GTP binding and G-alpha beta gamma heterotrimer dissociation. So one layer of our analysis in the lab to trying to understand how these different variants cause disease is to use functional assays in cells that monitor different aspects of G protein couple receptor signaling. This is, as Dr. Escobar described before, is the panel of our most recent cohort of mutants analyzed. And you can see from all the different colors that all these mutants show high levels of heterogeneity in their functional profile. But interestingly, all of them have this loss of function behavior, meaning that they show loss of GPCR signaling transduction. The loss of function behavior manifests through different mechanisms that include loss of expression, deficits in heterotrimer assembly, and deficits in receptor interaction. A subset of mutations show a dominant negative effect, meaning that the copy of the mutated protein interferes with the ability of the non-mutated protein to transduce GPCR signaling. And this also happens through different mechanisms, such as changing in beta-gamma coupling and changing in or gain in constitutive GPCR interaction. Then, as Dr. Escobar described before, we kind of define a severity score for the different mutations, and what we found is that all the mutations kind of fall on a continuous spectrum between loss of function and dominant negative activity, with a high correlation between the severity of the functional disruption that we observed in our assays. Our second level of analysis is to actually study the impact of these mutations in vivo, and for this we use mouse models. In this particular study, we used a viral strategy to overexpress two hotspot dominant negative mutations, the G203R and the R209C. And we focused on one prominent feature of the GNAO1 symptomatology, which is the movement disorder, and decided for this reason to target the striatum, which is a key brain structure controlling movement and coordination. And in fact, disruption in striatal activity leads to many movement disorders such as dystonia, Parkinson's, and Tourette. And importantly, other mutations in striatal-enriched genes such as ADCY5, G-beta1, GNAL, which are all implicated in GPCR signaling, and in particular in the cyclic MP axis, have been associated with motor, epileptic, and neurodevelopmental disorders. So in this study, again, we overexpress the two mutants or the wild-type G-alpha-O in two different populations of striatal neurons, and we found that overexpression of both mutation in either population of neurons result in dystonic behavior and overall loss of coordination and balance. Then for further model refining and development, we decided to focus on the G203R mutation, which is one of the most commonly found mutations found in patients with severe movement disorders and seizures manifestation. And importantly, there have been attempts to create GA2 mouse model carrying these mutations, but unfortunately, these attempts were unsuccessful due to the lethality of these mutations when it's expressed constitutively in mice. So to overcome this particular limitation, we devised a conditional genetic model in which the mutations, the G203R mutation is introduced in exon 6, in endogenous exon 6, but it's only expressed upon CRE-mediated recombination and deletion of an artificial cDNA cassette flanked by the two LOXP site that are recognized by the CRE recombinase enzyme, and that then switches the splicing to the endogenous exon 6 containing the mutation. The resulting mouse will have a copy of the wild-type allele and the copy of the knock-in or recombined allele that mimics the heterozygosity state of patients. Importantly, we also introduced a TD tomato fluorescent reporter in this line that allows for functional validation of the recombination strategy. So for introducing the mutation, we need a CRE driver line that actually drives the recombination, and for our initial studies, we decided to use a very well-validated RGS9 CRE driver line, which we used widely in the lab. And with the RGS9, we are able to achieve highly selective recombination within striatal neurons. Luckily, the mice were viable, fertile, so we could perform our characterization. We started by looking at TD tomato expression in striatal tissue. You can see, indeed, TD tomato is expressed in a variety of cell types, because I forgot to say it is under the control of an ubiquitous promoter. But upon CRE-mediated recombination driven by the RGS9 promoter, we see severe loss of signal that is, you can appreciate from this magnified picture, arises mostly from lack of signal in neuronal cell types. We perform further molecular analysis by looking at mRNA expression of the GNAO1 gene in the knock-in mice compared to the wild-type mice, either expressing or not the CRE recombinase, and found no changes in GNAO1 expression. We also checked that the mutation was actively transcribed into messenger RNA by RT-PCR sequencing. And by Western blot, we looked at the expression of the GAO protein and found no changes consistently with the mRNA study, no changes in GAO protein expression between knock-in and recombined mice. Then we extended our molecular analysis to more molecules that are important for striatal physiology and for GPCR signaling. And importantly, we found significant changes in the expression of cyclase type 5 and GALF, which are critical elements of the cyclic MP axis. And this is consistent, it's kind of in line with our previous evidence that G-alpha-ALF signaling interacts with cyclic MP axis. And this is also very specific because we didn't see changes in the expression of other striatal markers such as DARP32. So after this initial molecular validation of the model, we performed our behavioral analysis in mice using adult mice at about 8 weeks of age, where the recombination is supposed to be very well established. And for all the behavioral testing, we focused on two prominent symptoms of the GNAO1 G203R pathology, which is movement disorders and seizures. These are the results of the open field. So the mice are placed in the center of an arena and allowed to explore. And what we see is that the mutant mice show high hyperactivity in the testing arena, which is manifested as sustained forward locomotion after an initial period of acclimation and no changes in vertical activity. We then performed an accelerating rod-rod test. For those of you who don't know, the mice are placed on a rotating rod that goes at accelerating speed, and we monitor the time the mice are able to run on the rod. And to really point out the role of the striatum for movement disorder and G203R symptomatology, we found that the mutant mice show severe disruption of motor deficits and overall motor learning on the accelerating rod. For seizure susceptibility, we used the chemical convulsant PTZ model at doses that were not expected to cause severe seizure manifestation. And indeed, at the dose of 30 milligram per kilogram, only a minority of mice showed tonic-clonic seizure. And overall, we didn't find differences between mutant and control mice. Then to probe the impact of the mutation to several brain regions, we crossed the knock-in mice with different Cre driver lines. We used the nesting Cre to drive constitutive brain-wide expression of the mutations, of the G203R mutation, but unfortunately, and in line with previous evidence, mice were not viable. We used the CAMK2 alpha Cre driver mouse line to restrict the expression of the mutation in excitatory neurons of the forebrain, in particular cortex and hippocampus, which are known to be critically involved in seizure. And in this case, mice were viable. And however, I can tell from the first time I was trying to work with this line, they were kind of disrupted. Already in their home cage, they show jumping behavior, hyperactivity to handling, and importantly, spontaneous seizures. And most of all, we found that there is a steep decrease in the survival rate of this mouse line at around 8 weeks of age, with only 20% mouse viability at 3 months. We performed the same battery of behavioral testing in this mouse line, starting with the open field. You can see this strain of mice also show hyperactivity in the open field, which, however, you can appreciate this hyperactivity is manifested mostly during the first period of acclimation to the testing arena and eventually falls to the level of the wild type of the control mice. And the hyperactivity is shown as both forward and enhanced vertical activity. No changes were found on the CAMK-3-induced mice on the rotor rod performance. For seizure susceptibility, we used wider range of doses, given that these mice were already showing seizures at baseline. We started at 20 milligram per kilogram, a dose that is not expected to produce seizure manifestation in control. And indeed, we didn't find tonic-clonic seizures in control mice, but about 20% of the mutant mice showed severe seizures. This percentage raised to about 50% at the 30 milligram per kilogram dose, and at 40 milligram per kilogram, which is still considered a subthreshold dose for seizure induction, we found that all the mice showed severe seizure, with some of them eventually dying from respiratory arrest. So now that we have defined behavioral phenotype of these mice that mimic some of the symptoms of the disease, we want to test for therapeutics and see how we can restore this disrupted behavior. And among the long list of therapeutics that we wanted to test, we decided to start with caffeine, based on the very promising results from Dr. Martinelli's lab showing that caffeine is able to restore motor function in the C. elegans model of the disease. Caffeine consumption has also been shown to improve motor symptoms of Parkinson's disease and ADCY5-related dyskinesia. And with relevance to our striatal model, caffeine is an antagonist at A2A receptors, which are highly expressed in the striatum and are known to affect cyclic MP production. We started by testing caffeine on the RGS-9 driver line in the open field, and as expected, caffeine treatment results in enhancement of locomotor activity in control mice. But in the mutant mice, which are already hyperactive, we found this very strong paradoxical effect of caffeine in actually bringing down their locomotion. These are the overall results of the experiment, and you can see that mutant mice treated with caffeine are brought down to the levels of control mice treated with saline. We also tested the efficacy of caffeine on the motor dysfunction on the rotor rod, and saw trends to amelioration of the motor learning dysfunction, and we are performing more experiments to confirm this trend. So some take-home messages from today. Brain-wide expression of G203R is little. Expression in striatal neurons induces hyperlocomotion, disrupts motor behavior, but does not change susceptibility to seizures. If we confine the expression of the G203R mutation to excitatory neurons of the forebrain, we observed early lethality, hyperactivity, and increased susceptibility to seizure. Importantly, treatment with caffeine ameliorates the hyperkinetic behavior and improves motor learning in mice with striatal expression of G203R. Just to wrap up, what we have learned from today is that we have a novel genetic mouse model that is still under development, but it's now available. And this mouse model really mimics the core symptoms of the disease, and we think it can help a lot establish causal connections between brain circuitry and biology to GNAO1 disorder. We've also shown that this mouse model can be used for preclinical drug testing, and we really hope this can lead to a more targeted therapeutic approach for GNAO1 dominant negative and GNAO1 disorder in general. With this, I want to thank my PI, Dr. Kirill Martemyanov, for allowing me to be here today and really getting to know this GNAO1 community. And the people in the lab, in particular Grant, Kevin, Colton, Natalia, Nina, and undergrads Brandon and Nicholas, who really helped with in-depth discussion and with data acquisition. And of course, the Bell Foundation, the Child's Cure Genetic Research, and NIH for funding. Thank you all for the attention.
Thank you, Gloria. Questions?
Thank you for the great talk. The mouse model is fantastic, but I'm even more excited by the fact that you find a decrease in the GNAO expression in this mouse model, so which fits very well to what we find. But maybe you have some other explanations to that or any other speculations. I also find that the adenylyl cyclase levels are decreased, so can you explain why do you think this happens?
We have ideas. We are working on it, so I kind of throw it there, and hopefully we can confirm this with experiments. One thing I really have in mind is that by retaining the beta gamma on the G alpha subunit, maybe the cyclase is less stable, and therefore we see this reduction. But I really like your hypothesis of having a chaperone molecule that is stuck with a mutant protein, and therefore we have all these changes in other signaling pathways. Yeah.
Hi. Thanks for your talk. I was just wondering, do you have any clues why brain-wide expression is lethal? Do you know which region might be causing the lethality?
Okay, here I want to point out something that, for sake of time, I didn't really specify during the talk. Nestin-3 is embryonically expressed, so it's very likely that embryonic expression of G203R has to do with changes in neuronal development. The Cre driver lines that we use are postnatal. They are developmental. The expression of both RGS9 and CAMK Cre raise up postnatal and establish at about P15, both of them. So that's the reason why, more than brain region, I think it's also time dependent.
So they don't make it at birth, or
the nesting? No,
okay.
I don't have much evidence, but I've been trying to genotype the pups right at P0, and I haven't been able to find a single mouse with carrying the mutation.
Yeah, thanks.
If there are no, one more question.
I just want to add to that nesting thing. We tried, and I think Vladimir, you also tried, right? We tried to create like regular CRISPR mice, not the knock-in ones, and we couldn't get the pups to actually survive. And even if they would, they'd probably die very early. So maybe that explains it.
Mm-hmm.
All right, thank you very much, Gloria.